cell membrane is a semi-permeable membrane surrounding the cytoplasm of the cell. It separates the interior of cells to the outside environment. The cell membrane is selectively permeable and controls the movement of substances in and out of cells. It consists of phospholipid bilayer, proteins, carbohydrate chains, phospholipids, and transport proteins. It's involved in cell adhesion, conductivity, and cell signaling. The fluid mosaic model of the membrane explains various observations regarding structure of functional cell membranes. It describes cell membranes and dimensional liquid and the two-dimensional liquid where phospholipid and protein molecules diffuse easily. The main membrane is composed of phospholipid molecules, integral proteins that are integrated completely into membrane system structure with hydrophobic membranes spanning regions interacting with hydrophobic region of phospholipid bilayer. And lastly, the membrane proteins are membrane receptors that relay signals between cells' internal and external environments. The transport proteins move molecules and ions across the membranes.
Facilitated transport, also known as facilitated diffusion. Materials diffuse across the plasma membrane and down their concentration gradient with the help of membrane proteins. A concentration gradient allows them to diffuse into the cell without using energy. These materials are ions that are polar and are repelled by the hydrophobic inside or middle of the phospholipid membrane. The material attaches to protein receptors on the membrane surface and are passed to specific integral proteins that aid their movement. Channel proteins are specific for the substance that is being transported. They're exposed to the inside and outside of the cell and have a hydrophilic channel through the center that allows polar compounds to enter the membrane. Aquaporins are channel proteins that allow water to pass through. Channel proteins are either always open or gated/controlled. Carrier proteins bind a substance and change its own shape, moving the substance from outside the cell to the inside and are specific for a single substance. Carrier proteins transport more slowly than channel proteins. Active transport. Active transport requires energy in the form of ATP to move a substance against its concentration gradient. Primary active transport moves ions across a membrane, creating an electrochemical gradient, for example, the sodium-potassium pump. Secondary active transport, also known as cotransport, uses the electrochemical gradient created from primary active transport to transport other substances into the cell, for example, amino acids and glucose. The nucleus. The nucleus houses the cell's DNA and directs synthesis of ribosomes and proteins. The nuclear envelope is the double membrane, phospholipid bilayer structure that forms the outside portion of the nucleus. This contains pores that control the passage of ions, molecules, and RNA between the nucleoplasm, or the fluid in the nucleus, and cytoplasm. Chromosomes are the structures in the nucleus made of DNA, which is linear in eukaryotes. Chromatin is the structure that forms when proteins are bound to a chromosome. The nucleolus is the darkly stained area in the nucleus that congregates RNA with associated proteins to assemble ribosomal subunits that are then transported out through the pores in the nuclear envelope to the cytoplasm for translation.
Endoplasmic reticulum is composed of two types, which is the rough ER and the smooth ER. The rough ER is surrounded by ribosomes, and it's the site where protein synthesis takes place. The smooth ER is not surrounded by ribosomes and is where the metabolism process takes place. Golgi apparatus is membrane-bound organelles, and its main function is to modify, sort, and package macromolecules that are synthesized by cells for secretion. Vesicle is a small membrane-bound sac that stores and transfers substances throughout the cell.
Lysosomes. Lysosome is an organelle in the cytoplasm that involves digestion and the waste removal throughout the cell. Lysosomes are organelles that contain digestive enzymes, and they digest excess or one-not organelles, food particles, and engulf viruses or bacteria. Peroxisomes are an organelle that is in the mitochondria. It is found in both animal and plant cells. It has a couple of different functions. It includes lipid metabolism and chemical deoxification. The major function of the peroxisome is to break down very long fatty acid chains through beta oxidation. In animal cells, the long fatty acid chains are broken down into medium fatty acid chains and are shuttled into the mitochondria and then are eventually broken down into carbon dioxide and water. Centrioles. Centrioles are organelles in the cell. They are only found in animal cells. The main function of a centriole is to help with cell division. Centrioles formate their spindle fibers to separate chromosomes during cell division and mitosis. Centrioles are protein strand microtubules. They are made of 9 triplets of microtubules in the shape of a cylinder. They make the centrosome, cilia, and flagellum.
The cytoskeleton is a structure that helps cells maintain the cell shape and internal organization, and also provides mechanical support that enables cells to carry out essential functions like division and movement. There is no single cytoskeleton component. Rather, several different components work together to form the cytoskeleton. The cytoskeleton of eukaryotic cells is made of filamentous proteins, and it provides mechanical support to the cell and its cytoplasmic constituents. All cytoskeletons consist of 3 major classes of elements that differ in size and in protein composition. Microtubules are the largest type of filament with a diameter of about 25 nanometers, and they are composed of a protein called tubulin. Actin filaments are the smallest type with a diameter of only about 6 nanometers, and they are made of a protein called actin. Intermediate filaments, as their name suggests, are mid-sized with a diameter of about 10 nanometers. Unlike actin filaments and microtubules, intermediate filaments are constructed from a number of different subunit proteins, as you can see in the PowerPoint, as a structure of the cytoskeleton and the components of it. Mitochondria are known as the powerhouse of the cell. They are organelles that act like a digestive system, which takes in nutrients, breaks them down, and creates energy-rich molecules for the cell. The biochemical processes of the cell are known as cellular respiration. Many of the reactions involved in cellular respiration happen in the mitochondria. Mitochondria are the working organelles that keep the cell full of energy. Mitochondria are small organelles floating free throughout the cell. Some cells have several thousand mitochondria while others have none. Muscle cells need a lot of energy, so they have loads of mitochondria. Neurons don't need as many. If a cell feels it is not getting enough energy to survive, more mitochondria can be created. Sometimes the mitochondria can grow larger or combine with other mitochondria. It all depends on the needs of the cell. Mitochondria are shaped perfectly to maximize their productivity. They are made of 2 membranes. The outer membrane covers the organelle and contains it like a skin. The inner membrane folds around many times and creates layered structures called cristae. The fluid contained in the mitochondria is called the matrix. As you can see in the PowerPoint, there's a structure of the mitochondria. The endosymbiotic theory describes how large a host cell and ingested bacteria could easily become dependent on one another for survival, resulting in a permanent relationship. Over millions of years of evolution, mitochondria and chloroplasts have become more specialized, and today they cannot live outside the cell.